Hello and welcome to the talk through of the mock. I'll show some of the mock scheme out as well because I can't write everything in my answers that's on a mock scheme. Um, so hopefully the aim of this is that I can just go through a few tips as I go through, why I put the answer, etc. So 1.1, yeah, you just need to know, this is just knowledge. Yeah, chloroplasts have have DNA and therefore they also have ribosomes, they have 70S ribosomes, so yeah, transcription and translation will occur in chloroplasts, and it's going to obviously transcription occurs in the nucleus of a cell. So that's just knowledge based, or you can get it by elimination. You just say two locations, so it's definitely not cytoplasm, because that's where translation occurs. Ribosomes, it's where translation occurs, and permanent vacuole has nothing to do with it. It says a plant cell, so it's not a bacterium, so yeah, it can't be cytoplasm. And then 1.2, hopefully you know this from your topic 8 stuff anyway. And I probably, again, it's worth looking at the detail that I put in the answer because I never know what's on a mark scheme, so you never know how much detail to put. So if you compare my answer to the mark scheme, I probably put too much for the 3 marks, but I might have got a 4th mark if it was possible. I'm really careful with bases on free nucleotides. Yeah, I don't want to make it sound like a nucleotide is binding. It's the base on the nucleotide that forms hydrogen bonds with the exposed bases on the antisense strand of DNA. So I'm always really careful on that. And then I always give an example as well. It's always worth giving an example. And I always pick the adenine uracil thing. It really does show to the examiner that you know what you're talking about. So yeah, if adenine is exposed on the DNA, then it'll be a free nucleotide with the uracil as its base that will bind and form hydrogen bonds. I could have possibly combined those two sentences to make it easier. And you can do this in the exam, like bullet point, like I'm doing here. And then, yeah, names of enzymes get marks. So always, yeah, names of enzymes, names of bonds. I mentioned hydrogen bonds. RNA polymerase moves along this antisense strand and it forms phosphodiester bonds between the RNA nucleotides and in a condensation reaction, just in case that is worth a mark as well. And you can prepare for that question because it's like a stock answer. So that's that one. Let's have a look at this next one. And this is just telling us that that exons are cut out. It doesn't say splicing, so my spidey sense is thinking, that an answer might be splicing, but it turned out there wasn't. But as I read it through it, I thought that. So yeah, each molecule contains some of the original exons. So some of the exons are being joined together, some aren't. They could be in different orders. However, the sequence of exons in a strand will always be the same as in the original pre-mRNA. So 1.3, explain the function of the codon at the start. Yeah, so that's just where the ribosome binds. End of story. And then I've got a 3 marker. Come on, suggest why a variety of different protein structures could be formed from this polypeptide. This is as I always, again, I teach this at GCSE as well. I think I say SSS, so that's my GCSE version, sequence shape, which changes to S S S R S at A level, where the sequence of bases of a gene code for a sequence of codons in the mRNA that will result in a different sequence of amino acids, and due to their different R groups, you'll get a certain shape of protein, and I'll say tertiary structure rather than shape. Here, I'm starting with an mRNA molecule, so I can't get my first S into the answer, but it's only 3 marks anyway, and not 5. So I'm going to give my 4 mark answer instead. So I've got exons in a different order. There will be a different sequence of bases. Different tRNA anticodons bind to mRNA codons. I always get that in. Anticodon is a tRNA word, mRNA is a codon word. So a different sequence of amino acids will be joined. I could have also said that the tRNAs carry a specific amino acid, which I probably should have done, but I thought space was limited. And then because I've got different amino acids, I get to my R. Different R groups of amino acids mean I get different bonding occurs. So will result in a different tertiary structure. That's the same as my shape here. And give an example of a bond, disulfide ionic. I've gone for hydrogen bonds forming in different places. So yeah, that's the classic. It's a transcription translation question. Is this? And it's pretty much a stock answer. So yeah, so if I'm revising this, that's on, my SSSRS is at the top of my revision notes, and then I will just write out an SSSRS answer of why a gene can code for a different shape of protein. Often they ask this in terms of how could a mutation cause for a non-functional enzyme to be produced, in which case you can give an example of a mutation that changes the sequence of bases in the DNA. A deletion mutation might occur within an exon of a gene, and therefore you get a different sequence of mRNA codons because it's an addition mutation. That might cause a frame shift, therefore all the codons will be in a different sequence, and then I can get my anticodon-codon pairing, therefore amino acids are in a different sequence that have different R groups, different tertiary structure, and if it's an enzyme, I'd say a different active site, and therefore not complementary to substrate, and I'm basking in the glory of getting loads of marks. From protein to protein, question 2. Yeah, so it's the enzyme lysozyme, and near a covalent bond labeled between X, which links two cysteine amino acids. Well, they're the ones that contain sulfur, so it's disulfide crossbridge. That should be straightforward. It's a spiral is Y, so it's the alpha helix. I can put an alpha or right alpha. And lysozyme is found, in a phagocytosis. Yeah, so it's the lysosome. So yeah, just tricky, but knowledge based. There's no need to dwell on that. Again, if anything comes up that you didn't know, then that's really useful because it's you have time to get your head around it earlier. Use some DSP time. And then we've got this, again, it's quite nice. It's not in like a horrible context or anything, so there's not a lot to get my head around. Enzymes are affected by temperature. Yeah, the graph shows the course of a million enzyme reaction at different temperatures. And then explain why there's different in the shape of the curve at 37 and 60. Yeah, so 60 is steeper, 37 is shallower, 60 goes flat earlier, 37 goes flat later. So I just need to get the steeper bit and the earlier flat. So yeah, 60 degrees C, the initial rate, that's quite useful. Yeah, it's the the rate at the start, how steep it is at the start. It's faster, there's more kinetic energy, more successful collisions between enzyme and substrate, or more enzyme substrate complexes form in a given time. Could I put? And then, so that's my weights. And I have put faster because I'm comparing them. And at 60 degrees C, the enzymes denature faster. All enzymes are denatured earlier, so substrate is not all used up. Yeah, so it must have not, they must have denatured before it's complete because that one's gone higher. People think that enzymes denature at a temperature. Yeah, they denature. They'll be denaturing at 25 degrees C. It's just not likely. At 37 degrees C, it's more likely that they will denature. At 60 degrees C, it is highly likely that they will denature quickly, and therefore, yeah, you do get a fast initial rate of reaction, but it's going to tail off. That's why we can't sustain a fever for like days and days. At 40 degrees C, it'll be a faster rate of reaction, so everything in your body is happening faster, but you have to kind of like, also enzymes are denaturing faster, so you have to remake the enzyme, so it just wears the body out. So yeah, so you seek medical attention if you've got like a more than a fever for a couple of days. It becomes quite critical. 2.5, explain why there's a difference in the shape. Yes, it's just 25 and 37 now, so it's the opposite. So at 25 degrees C, there's less kinetic energy, so less chance of a successful collision, fewer enzyme substrate complexes form. And then by 60 minutes, again, there's a number on the graph, so I may as well use it because it's still increasing. It says 60 minutes here. Well, it's still going up is the 25 degree C one, whereas the 37 one's gone flat. So it's either they're either denatured at 37 or they've used up all the substrate. But yeah, the 25 degree C one is going to carry on going. Paramecium. It's a single-celled organism, lives in ponds. It's shown below. So I've got a contractile vacuole. So I might not have heard of that, and collecting channels. So it's going to tell me about it anyway. So most species of paramecium are freshwater organisms. Over a period of time, water from the cytoplasm is collected by the collecting channels, right? So these are the collecting channels. They pass the water into the contractile vacuole. So there's my contractile vacuole, right? Once the contractile vacuole is full, it contracts, expelling the contents from the cell. Explain why paramecium needs to expel water when in fresh water. So it's an osmosis question. So it must be to prevent it from bursting due to internal pressure. And then I want to give it an osmosis answer. So the paramecium cytoplasm has a more negative water potential than the surrounding fresh water. So that's my priority. I want to talk about water potential and osmosis than the surrounding fresh water. So water will enter the cell by osmosis. It's going to swell. If it doesn't get shut of it, it's going to burst. Then they carried out an experiment in which the frequency of the vacuole contraction was observed when bathed in different concentrations of salt. The results are shown in the table. So let's check the table for, so it's going 0, 1.3, 0.3, right? So it's not going up in equal amounts. And that's going down, 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 down. All right, I wasted my time, because sometimes it becomes important that, and sometimes you say, right, Ralph, well, that's a 0.3 difference, that's a 0.5 difference, but they're not going down in equal amounts anyway, so it's kind of pointless me doing that. So I'll probably look to the questions to see if it's saying describe, then I might come back and do some more work there. Explain why there's a decrease in the activity of the contractile vacuole as concentration of sodium chloride increases. So yeah, I don't have to say why. Sorry, I do have to say why. I don't have to say describe the data. It's quite boring, is the data anyway, as it's all showing the same trend. So I'm just going to say, yeah, as there's more salt, NaCl, so as the concentration of salt increases, the water potential decreases. Yeah, and just as a little tip there, pure water has got a water potential of 0 kPa, and as I add solutes, I get more negative values. So it's going to go to -3 kPa kilopascals if I add solutes to it. So the more solute you add, the more stuff that dissolves, the more negative the water potential becomes. I'm very careful with lower and higher for water potential. I know lower does mean more negative, but some people think that like -3 and -3.1 and -3.2. -3.2 is lower than -3.1, but it's a bigger number, and some people think it's a higher number. So again, I just stick to more negative. So I've got then the more negative water potential, and I can use my symbol for water potential of sodium chloride. The closer it comes to the water potential of the cell. So this cell is going to have. Let's go back to the data quickly. So it still loses. It's still having to contract this contractile vacuole at 0.2. I could plot a graph of this, yeah, and so I've got my sodium chloride concentration from 0. There's 0.2, and then I've got my mean number of contractions. It started at 6.5. It's decreasing. I could then extrapolate to here and get, all right, well, it was 0.25 NaCl gives me the same water potential as what's in the cell. If that was the case, it wouldn't have to do any contractions, because no water would move in, because no osmosis would be occurring. And that's what I'm trying to say here. So the more negative the water potential, the closer it comes to the top of the cell. This means that the diffusion gradient for water decreases. Less water enters the cell by osmosis. And as less water enters the cell, when it's more concentrated, outside, less water enters, so less needs to be removed. It does say, explain the decrease in activity of the contractile vacuole. So I do have to get to that point. That's where people can end up saying the first bit. Why is the contractile vacuole less active at higher concentrations of salt? Because less water is entering, therefore less needs to be removed. So you do need to get the removed bit, or a word that means the same thing. I think the Morse scheme says expelled. But but as a teacher, it's always a bit of a harsh word to use, expelled. And then the cytoplasm paramecium contains salt crystals. These can be used to alter the water potential, suggest and explain. So it suggests and explain. So it could be anything sensible, how the water potential can be altered and how this could benefit the paramecium. And that's crucial. I have to get to the benefit to the paramecium. Again, it's exam technique is this. But a lot of you haven't got to the benefit. But who knows? So I've just put, if a cell converts soluble salt into insoluble crystals, the crystal is visible, it's not dissolved, isn't it? Then the water potential in the cell will increase, it becomes less negative. So if I turned all the salt into a crystal, I'd go to 0 kPa. It would have no solutes in it, it would all be out of solution. So yeah, it's gonna raise the water potential, but yeah, less negative is less confusing. And then if I place a cell in fresh water, when it rains, so these live in fresh water. It's going to rain, and the water is going to get more pure as it dilutes whatever salts are in the actual pond to start with. So yeah, less water will enter by osmosis. So if a cell is closer to 0 and the water is 0, then that's less water entering, therefore less energy required to power the contractile vesicles. It's contracting, so it's going to be using ATP. So again, that's my benefit bit. Therefore, less energy is required. It's often comes up so often. Almost everything is to do with energy. Like why is this? Yeah, it'll go down to being some kind of energy efficiency type thing. So it's often a good tip to think about. We're almost there. Question 4, knowledge. So yeah, ribulose bisphosphate, biphosphate, triose phosphate. For B, glucose goes to polysaccharides, and enzymes are made of amino acids. So it's D. Again, should have been easy. Then we've got this load of detail. Where's my highlighter pen? Let me just get my highlighter pen going to show you how I'll do it. So they're adding 200 centimeters cubed distilled water. So that's a control variable. They're going to add in 5 grams of sodium hydrogen carbonate in excess. So there's an excess of CO2, right? So that's not a limiting factor, it's CO2. They're going to place it in a water bath at 10 degrees C. I'm just going to put that in red because as I see here over here, that's my range, so that's my dependent variable. Leave it for 10 minutes, that's a control variable. Insert an oxygen sensor into water and measure the baseline oxygen concentration. Okay, place 100 grams of this species of is it algae, pondweed, sorry, into the beaker. Remove all of the light sources, that's another control variable. LED light source, so there's no heat, 20 centimeters, control variable. At the top of the beaker, control variable. Use a light intensity meter to measure the light intensity above the beaker is 5,000 lux. Okay, control variable. Measure the concentration of oxygen, right? So that's my dependent variable. That's what I'm measuring is oxygen concentration. And I'm doing that every 10 minutes for 200 minutes. That is not a control variable. How often you record something is not a control variable. Carry out 4 more repeats at 10 degrees C. So yeah, that's my, again, not a control variable. That's for my, so my mean is reliable. I can spot anomalies, etc. It'd be better if it was a numbered list, but they've tried. So yeah, independent variable is temperature. I've already cottoned onto that because that's what we are varying. And then identify one variable that is not controlled in this one. They've controlled blooming everything. So yeah, if it's in water, you can always put pH. Top tip, always put pH. The other one you can always have in biology, because we've got a living thing. I know we've got a certain mass of this pondweed. I've got 100 grams of it, but it can be the same age. I can't put same species because it's given me its genus and species name, but I can say like same age, same surface area, anything like that. Yeah, something about the living thing. So if an experiment is in solution, you can always control pH. I say always, not sometimes. If you are, well, often it's the dependent variable anyway when they're measuring pH in like a lipase experiment where it's making fatty acids. And last two, 4.4. Again, it looks complicated, this, but it's actually quite simple. It's just making sure that you know, like I like checking the lines really. So 10 degrees C, all right, well that's that one. 15 degrees C, well that's that one. 20 degrees C, that's that one. 25 is that one, and 25 and 30 is that right. So it is. There's no tricks here. It's just like as temperature increases, the concentration of oxygen measured increases. It's describe and explain. It's only worth 3 marks, so it can only be. It's going to be a 1-2 split. It could be a 2-2 split. If it was 4 marks, I would write about this area here. Yeah, that if I was zooming to the start, can you see how that they're like the same there and they're the same, and then they gradually kind of disappear. There's a slight wobble here. I'll be getting into more detail about that kind of like nuance of difference. Um, but because it's a 3 marker, there has to be more marks in the explanation. And then yeah, as it's warmer, the enzymes in photosynthesis work faster, so photolysis of water will be faster. Therefore, more oxygen is produced. It's just what makes oxygen in photosynthesis? Well, it's the photolysis of water. And then light-independent reaction it can be referred to as the dark reaction, not because it's sinister, because it happens in the dark. Explain why this is both accurate and inaccurate. Well, it's accurate because light is not directly needed for Calvin cycle, and it's inaccurate because, and then it's like, well, there's that step of Calvin cycle or the light independent stage or the LAR that needs that reduces GP into TP, and that requires NADPH and ATP, and they come from the light dependent stage. So it's a noise question, and I know it's a noise question because again it says explain. So there we go. Hopefully you found something useful there. Again, I'm trying to keep these short, so I'm like about 20 minutes, so that's not bad. Good. If you like this video, click the like button. It's not one to like.